Vanessa, I want to stick with you to talk a little bit about barrier systems and
Ooh.
isolators for small molecule drug manufacturing. What maybe you could tell us what role barrier systems play and perhaps how restricted access barrier systems and isolators are used effectively.
Yeah, so I love barrier systems. I think that if you read EU Annex 1, you see if you read between the lines, if you understand, the intent as well as the plain language that's in the guidance, it's very clear that barrier systems is where you need to be. So if you're constructing new facilities, the expectation is you have them. And you have isolators is obviously the preference if it makes sense to build your process in an isolator. I think also the expectation for older lines or aging facilities is that you do a retrofit for a barrier system that makes sense to your filling line. And the whole principle is very simple. It's to provide a barrier, a separation between people, the operators, and the product. It's a physical layer of protection to reduce particulate, chemical, air, whatever contaminations coming from the operator, the people, right? Number one in the room, but also the background area. So for small molecules, for these types of products, I think it depends on the type of small molecule you have. So barrier systems can be for containment purposes, right? Especially if you've got powders or if you're compounding APIs or something like that. They can help contain while also simultaneously protecting, obviously, the operator, but cleanliness of that, that compounding step, if you will. When you're talking towards fill finish, barrier systems really provide a microbial contamination control unlike anything else. So for isolators, it allows you to decontaminate the area, right? Using a validated process. Even some closed RAB systems, there's new technology out there that's also letting you decontaminate them. I haven't worked with it yet, but it's really encouraging and exciting. But even closed RABs or even open RABs, you can clean those systems, let the area recover prior to doing your actual fill finish. So they give you a lot of protection for the product. I think that's why we're in this business, is to deliver products to patients, right? Not just to formulate, but they reduce contamination risks. I think that they're an expectation from regulators, but they play a huge role and a different type of role depending on the product and the process that you have. Yeah.
What about the differences between, say, open versus closed RABs, open versus closed isolators? Are there differing levels of sterility assurance associated with those different systems?
100%. I always say we just actually gave an entire webinar on this concept. You can look at it as like a spectrum, okay? And it's important that you understand that not all barrier systems are created equal, okay? And all barrier systems have vulnerabilities in them, okay? So open RAPs are just like that. They're open. They should be opened only under very rare or specific conditions in which the line otherwise wasn't retrofitted or wasn't designed properly, and you have to get equipment in or you have to do a specific intervention. Under those conditions, that's probably, it does provide sterility assurance, but it's not to the same degree as some of the more closed systems, right? It's quite logical. And while you're implementing those controls, if you do have to open the RABs, you're isolating product, you're probably, quarantining a certain portion of the product because you're opening it, exposing it to a background condition. If you go towards closed RABs, that's even better. That means the door is never open and you're operating strictly through glove ports. But understanding glove ports are not perfect. They're not, things do go wrong. Leaks, pinhole leaks, they're not installed properly or they're not disinfected properly. That's really important. And so, while you don't have people's gloved and garment hands in them, you do have them through glove ports, and those need to be managed properly. And obviously the aseptic interventions need to be done properly as well. Just because a lot of people think, oh, well, it's a sterile glove. It's not. It's not sterile. The area inside of RABs is not sterile. The area inside an isolator is not sterile, okay? It can be sterilized, and at a moment in time, you can say everything in there is surface sterilized. But the moment you introduce materials, air, it's no longer sterile. And then as you go from a closed RABs to an isolator, Isolators have glove ports too. The newest technology with the greatest degree of sterility assurance are gloveless isolators. Mm-hmm. So you can see that if you go from open wraps to closed wraps to isolators to gloveless isolators, you get an increase in sterility assurance, and you have better controls, and you reduce the impact of surrounding, operators, bacteria coming from people gowning and stuff like that. So I think it's important to know what type of barrier system you have, how you can decontaminate, and how you should perform aseptic operations in there. And then, we don't have enough time, but we can talk about robotics too. They give you a whole different level of, control in the way that you are designing the intervention to minimize, airflow disruptions and movements and slow and deliberate, these types of principles. So lots to be said about barrier systems. I think I've become like a fan of barrier systems, talking about them in my recent years. So.
But there's one aspect, a lot of folks have old lines. They put open wraps around them and they say, oh, we're good. And what we've seen over the years is then their air is being supplied by the room, and they don't go all the way up to the HEPA filters in the room. You've got to go and made up to that. But the other big issue is the HVAC in the room controls the contours of the air within inside that wraps. Mm-hmm. And part of the problem is we've seen cases time after time where the airflow in the room there are 3 returns on one side and 1 on this side, and the air is being pulled in a particular direction. And what happens is you have half of the line or half of that area is not even being covered by the clean air. We've seen situations where the air is on a diagonal and actually generates a swirling vortex in the middle of the wraps and actually suspends particulates up and holds them in the wraps and doesn't remove them. So if you're going to look at an old line and you're going to, just throw wraps on top of it, you have to completely look at the HVAC in that particular room. You've got to do a computational flow dynamic model of the air inside that and really understand, do you have the HVAC coverage? So I think that's one of the most important things that we've come across, and we see a lot of fallacies with, open wraps or even wraps that are active, but the bottom end of them are open. They have the separate air, but if the bottoms are open, it's very much dependent upon the HVAC in the room and opening and closing of doors. So we really got to focus ourselves on closed active wraps. We got to go to isolators, and I'm a big fan, and I bought a couple of the gloveless isolators, and they're the best.
Yeah, I think can we address that question? Oh, go ahead, Liz.
Oh, sorry, Vanessa. I was just going to say, I think what both Vanessa and Herman are saying is, understanding the limitations of these barrier systems. And again, my favorite thing is training and making sure that people understand the why, and having the right information to be able to make the right decisions for behavior and actions. So something like an airflow visualization study, which Annex 1 references as being part of the training program for any aseptic operators, so that you do know what the air is doing inside that barrier system. And your controls are, the amount of control that you can get from a barrier system is only as good as your inputs, whether that's something like the development of a VHP cycle or something like training to make sure that when your operators are physically putting, their gloved hands into a barrier system, they understand what they should be doing. And if you have a really, really, really great isolator design and a perfect VHP cycle, but your operators aren't behaving the right way with that isolator, it's not a magic box, right? And I think a lot of people are sometimes like, oh, it's the isolator, it's VHP'd, everything is sterile, and that's just not true.
Yeah, and because operators don't consistently deal with inside the isolator, so they'll pile up tools, they'll pile up disposables, they'll have their EM plates will be piled up there, they'll tear, and they'll pile up all sorts of stuff all over, sometimes obstructing air, just sometimes not being very tidy in there, and they don't dump it off into the dump spot in the isolator. So there's a lot of watching and understanding of the behaviors inside the isolator. And the other thing is, before the visualization studies, rather than burning all the visualization studies and putting all that time, do a CFD, do a computational flow dynamic model. You put it on your computer, run it, and if it makes sense, great, then do the visualization. But don't just go ahead and just say, oh, I think it'll be just fine, and go ahead and fail the visualizations.
Yeah, I think that what the take home from this conversation is that not all barrier systems are created equal. The barrier system that you have for your product filling line needs to be fit for purpose, for your purpose, right? It needs to consider how you operate. And let me tell you, if Herman, like how he described, if you've got buildup of tools, or if you've got glove ports that are in the wrong position, or when they're in rest, they're touching a line or something like that, that's a design issue.
Correct.
I can't fix that in my CCS, okay? So that's a redesign issue. That's working from a microbiologist along with an engineer to change the position of the glove port. You do that during a shutdown. Don't just deal with it, right? You've got to make those changes, because small changes give you big yields when it comes to, you know, grade A conditions. And I think that we underestimate the value in working with engineers to get redesigns done, even on isolators. Yes, they cost money, but you know what? We learn more about the process when we run it 100 times, as opposed to just some of the design spec conversations initially.